right, hi, my name's Janie Merkel and I direct the biology team at the Yale Center for Molecular Discovery. I've just started with a screenshot of our web page. We have a lot of information there, but it certainly doesn't substitute for talking to someone about your specific needs. So my last slide, I have my contact information and I have cards as well for anyone who can stick around. So I'm really happy to come meet with you and learn about what your needs are in early stage drug discovery or target identification using siRNA or speak by phone to help you learn what's possible for your project. I'm here today representing our team. We have 8 scientists and part-time administrative and lab support. And as our faculty director, Craig Cruz, likes to say, we are industry-trained scientists doing industry-style research, i.e. drug discovery research, in an industry-like setting. We are located on Yale's West Campus. But the piece that I think is really critical to add is that we're really meeting very innovative and individualized needs for Yale faculty. So we are looking at a huge range, vast biology. We do a lot of work in cancer, but we do a lot of work in many other fields. We work with faculty from over 25 different departments. So we do biochemical assays like enzymatic assays, protein-protein interaction. We do a lot of cell-based work, both animal and human cell line work, image-based assays, and we work even on whole organisms. Our libraries, our small molecule collections are in 384 well plates. So we're using small organisms like nematodes or algae or pathogenic bacteria or yeast or fungi, all sorts of things, but typically quite small. We can go to 96 well format if we have to. We actually had a summer intern doing 3D spheroids this summer in 384 well plates. We have a lot of resources, and given the time, I'm just going to go over them at a very high level, but again, I'd be happy to talk with you individually as these resources might relate to what you're trying to access. And we're all here today because of technology needs or resource needs. The way we work with people is we work very closely with you. It's a staff-assisted model, so once we understand what your needs are, you're paired with someone at the center, so maybe you or grad student or postdoc is working with that center member through all phases of your project. We're managing the technology, the instruments, the compound collections, the statistical analysis, and you're really experts on the biology. So the reagents we're using are yours, and they're different for every project, and that's what makes them special. And in terms of being challenging and successful, we're successful because you're really engaged, and you have so much knowledge about those reagents that maybe you developed. So we want to make sure we don't let go of that, and that's why we want you to be very highly involved. So we do assay development. We work directly in 384 well plates so that we're in the format we'd be doing screening. For screening, we do both small molecule screening. We have over 150,000 small molecules, and we do siRNA screening. We have a human genome collection. So that would be human cell-based assays. For the small molecule screening, again, it could be biochemical, cell-based, or organism-based. We do image-based assays, so we have a fairly high throughput fluorescence imager. It's four color fluorescence, wide field instrument, and typically we're doing this with fixed cells, and then we can extract and quantify features from those images that we use. So we're converting the complex images to numerical numbers, percentage of cells with signal over intensity or co-localized signal or any number of things. So we're developing those image analysis algorithms with you for your data. We have a lot of liquid handlers, so we've worked with people who just for one project might need to transfer the materials into 96 well plates or 384 well plates or 1536 well plates. Please don't buy a liquid handler for something like that. We really are happy to help you with that. So maybe you want to change formats or make replicates or make dilutions or anything else. We're happy to do that. We have sterile systems inside a biosafety cabinet, so if they're plasmids or something that you'll need to keep sterile, obviously we have the capacity to do that. We have a great team of chemists. They're excellent at making compounds, but they're also just excellent at finding compounds from reputable sources. So If you've tried to purchase antibodies and you know how variable vendors are, it's the same deal with chemicals. And so if you just want to buy something and you're not sure that you can't find it, maybe they can find it, or you don't know who to buy it from, they can point you in that direction. And if you can't find it, maybe it's something in the literature that someone's not going to share. They're happy to make it for you and give you a sense, once they know what the molecule looks like, how easy that would be to make for you or how challenging. They also do medicinal chemistry, that's changing molecules for desired properties like solubility or selectivity that's specific to your system. And that's very customized work. Sometimes they do the synthesis, sometimes they develop the plan and we outsource it to local contract research organizations because it's more cost effective for you when we do it that way often. They do a lot of computer modeling as well. So if you have a crystal structure co-crystallized with a substrate, they can help you identify new molecules that could take advantage of key groups on your protein to increase specificity or binding affinity or something else. It's based on the structure of your target. Molecular probe design is really about tool compounds. If you want to modulate the biology of your system with a small molecule, there are, depending on the context in which you're using it, considerations that would go into that. We do a lot of grant support, so letters of support, boilerplate text about screening, the resources we have. Again, we're really very happy to do that. We do many of those a year. Our chemists weigh in on intellectual property evaluations for people filing patents with the Office of Cooperative Research. They have the abilities and skills to do that and look at competitive landscape if you have a new small molecule. And we do various educational initiatives. One, which we'll be hosting on November 5th, is a 5-hour nano course where we teach you about biochemical assays, cell-based assays, and medicinal chemistry. It's fairly introductory in small group format, but it's a starting point for a lot of people who are thinking about doing a screen to get a sense of what's involved. I'm going to just spend the last 2 slides talking about Pitch. This is a new program that we've just recently received funding for from the state of Connecticut, specifically Connecticut Innovations, which is the venture arm of the state, through the Connecticut Bioscience Innovation Fund. This is a $10 million grant for 3 years, and it's a joint program between both Yale and UConn. The point of the grant is to generate new Connecticut-based startup companies specifically around new therapies. So if you have an idea for a new drug, we'd love for you to submit an application. You might not be ready this year, but you might be ready next year. So I just want to put that out there to you. It's really about translating what you're doing that's innovative into something that could really help people. We have a separate website for that, and I have some cards for that as well. www.pitch.yale.edu. Our next applications are due January 4th, so we'll be reviewing them every quarter. The real benefit to you is free services and expertise. It's paid for by the program. And the idea is really to modulate targets that impact disease. So it does have to be closely linked to disease. There are resources at UConn Health and Farmington in protein expression and purification. The center will be doing assay development and design and small molecule screening, and then medicinal chemistry will happen jointly at our center and at UConn School of Pharmacy and Stores. We have an external advisory board. These are non-Yale people. These are venture capitalists. These are serial entrepreneurs who have experience as founders of biotech companies who are evaluating the applications. They're making the decision on who will get funded by this program and who won't. There are 2 different entry points, so if you don't have an assay done yet, basically one entry point is at assay submission. A second entry point, if you have a small molecule but you need to optimize it, is in medicinal chemistry. So they're basically 2 stage gates, if you will. And the outcome is really presentations to venture capitalists. In addition to this external advisory board who have real working knowledge and expertise about what it takes to really make a new company, we will have a non-voting board of venture capitalists who will also be asking questions and providing insight into how to further strengthen these data packages. That's the point of this program, is develop what are called data packages for the venture capitalists. They are under CDAs, so people can include confidential information. And we're really excited about this initiative, but we need you to participate too. The application's a little unusual. It's an 11-point PowerPoint slide deck where you basically fill in the blanks. And here are just 3 of the slides. One of them is your name and the title of your project. For each PowerPoint slide, in the notes section, there are tips and facts for each slide to help you answer the questions if you're a little unclear about it. But again, I'm happy to answer any questions people may have about that or have about it as you consider filling one out or start filling it out. So, please do contact me if you have questions about assays or screening or the pitch program. My counterpart in chemistry is Denton Hoyer. He is happy to talk to you about synthesis, and I think this got cut off, computational modeling, virtual screening, and silico screening. We are located at the West Campus as well, so we're also happy to have you come visit us and give you a tour if that's useful. So thank you, that's all I have.